Healthy lakes and rivers. They add to the quality of our lives in Montana. They're a big part of why people come to visit and ultimately support our communities. Our waters also support a thriving agricultural economy. Whether you're an angler, a boater, or a rancher, aquatic invasive species are threatening the clean, clear waters that you and your fellow Montanans value and depend on. Monitoring for aquatic invasives across a state as large as Montana is a huge task and requires a lot of help from volunteers and watershed groups. Your willingness to help is valuable and very appreciated. This video will help you in your efforts to monitor your local watersheds. Aquatic invasive species come in all shapes and sizes. The list of invasive species threatening Montana's waters includes snails, mussels, plants, amphibians, fish, crayfish, and more. Despite this diversity, they all have a few things in common. Most of them outcompete native species. They also negatively impact the food chain or amount of oxygen in the water. If that wasn't enough, some also choke out sunlight and clog irrigation pipes. These impacts can be catastrophic, both environmentally and financially. However, if detected early, we have a chance to remove them, or at least stop them from spreading further. If we want to protect Montana's waters and everything they provide, we have to be vigilant, and that's where you come in. So we're glad you want to help. Now, let's talk sampling. First, it is important to understand that each of these species occupies their own niche in lakes and rivers, and this affects how to best monitor for them. For example, some live out in the water column, others on rocks and logs. Some are mobile, others are not. Some are large and quite visible, while others are tiny and nearly invisible. To be efficient with your limited time and resources, it helps to target a few species and to be strategic about how and where you search. So, let's get started. Plant invasions typically begin in shallow areas where people access the water. Here, a throw rake is the best tool.
Just essentially, it's a thatch rake that you can buy at any local hardware store. You cut the handle off, attach a rope to it. It's double-edged, which works really well because it doesn't matter which way it falls in the water when you throw it. And then you pull it through the water column slowly, and it will just grab onto any plants, and you pull them in. It's really important to keep kind of constant pressure on it as you're pulling it in, because otherwise plants start to fall off. It's probably the best tool, especially in waters that are a little more cloudy, you can't see. If you're sampling pretty shallow water, you can leave a handle on or get one with an extension on, and you can rake it that way as well. It's got two edges. We grind off the tip so they don't cut you. Or your boat, or scratch up your boat or whatever. It's really useful to use braided line over twisted line because it gets all twisted up as you're pulling it in. It's a simple tool, less than $30, $40. You have a tool that you can go sample for plants everywhere.
When water visibility is good, nothing is better than getting in the water to look around.
Snorkeling can be used pretty much for any sort of species that you're looking for. I mean, the beauty of it is you're getting in the water, you're cutting the glare out from just being on the surface. You can see everything moving around. You can see the plants. You can flip rocks while you're underwater. You can get up underneath a dock and see underneath for potential mussels attaching to those surfaces.
What'd you get?
If the water's clear enough, you can cover a large area fairly quickly and looking for everything. Flippers and fins and a snorkel is really all you need.
While it may be hard to spot a single snail or clam hidden in sediment, using a kick net can concentrate debris so you can quickly sort through it.
So this is a kick net. This is really useful to look for that stuff along the sediment. So essentially all you're going to do with this is you're going to, rake it through the trees or through under the docks, through the rocks and things like that. Dump it out into a tub, and then you kind of sift through it and see what you find.
There we go, yeah. And there it is, right
Kick nets are great to get into places, whether it's a lake or a river. You can use it in either places. You can get up under vegetation, scrape the sediments on the bottoms, and really be able to capture things that you wouldn't see otherwise. You know, there's a lot of different organisms and species on the sediment that are small, and you don't really see them, and they're a little cryptic until you can stir them up and get them into a tub to look at. So, you know, they run anywhere from $200 to $300 or more, depending on how fancy you get.
Yeah.
So this is one you can be pretty aggressive with and really go for it. Just if you just Google kick net, you can usually find some sites.
One of the easiest ways to monitor for mussels is to hang an object designed specifically for them to settle on.
So this is an example of a substrate sampler. There's lots of different types and ones that are available. This one is just bored essentially with a bolt that you hang underneath a dock or under a buoy or something. You leave it underwater. You could check it periodically, once a month or something throughout the season, to see if any mussels established on it, and then you can reuse this multiple years. It's a great opportunity for people that have docks or something to get involved.
Seeing small mussels attached to a surface is relatively easy. However, mussels present a unique early detection challenge because their life cycle includes a microscopic free-floating stage.
Okay, so this is the plankton net. So this is what we use to pull through to collect plankton out of the water column. Plankton are just the small plants and animals that live within the water column. Some of them you can see, some of them you can't see. So with those plankton, that's what you're looking for, is the veliger stage of the mussels. The veliger is just the larval stage of the mussels. So they reproduce in the water column, and these little mussels start to form, and they actually are light enough that they float in the water column. As they get bigger, they get it dense enough that they can actually settle down onto the sediments. So what we're looking for is that stage before they're too heavy and too big. This net will actually just grab those as they're suspended in the water column, and you end up with a bottle with all the plankton in it, and you can see stuff moving around in it, but it really takes someone looking at it under a microscope, dissecting scope, to actually find what we're looking for. You know, FWP has a lab that we get samples from people, and we process them for free of charge, because we're working as a group to really look for these things. So yeah, as simple as getting us those samples. In the lab, what they're doing is they're filtering out water, they're filtering everything too small out and everything too big out. And so then when they put them into the dissecting scope, they actually use a cross-polarized light. And what happens is it actually makes those shells glow into like kind of a cross, which makes them a lot easier to see through all the sediment and algae and other things in the water. If you're looking to send in plankton samples, it's really as simple as reaching out to our lab so we know they're coming, boxing up those bottles so they won't leak, and sending them into the lab. And we have a form that you can fill out, and we'll let you know what we find. The information that you want to include on all of these is, water, obviously, what name it is, latitude, longitude, maybe some habitat, the date, collector, who collected it. The basic information of, we're not there to collect those samples, so the more information you can provide us to paint a picture of what it looks like at that habitat is better. We do return your bottles if you're shortchanged and you don't have enough money to buy new ones every year. So that is something you can reuse potentially for a couple years, so they are reusable. So there's typically 63 micron, which is the size of the mesh material. There are different sizes that you can get. This one is a 50 centimeter. It just collects more water. There's smaller ones that are a little easier to store as well. But essentially, you pull it through the water column and it ends up here in this end, and you can condense it all, and then you can unscrew it and dump it into your bottle and then preserve your sample. So they're pretty fragile, so you got to take care of them when you're out and about. They're easy to snag on things, so you just kind of watch where you're pulling them through, and they'll last for years. Yeah, these aren't cheap. They start at $300 or $400 and can go up depending on the size you get. So we take care of them. When we carry them, we actually put them in their own special case, put a tube sock on the end to protect the screening, kind of treat them like a baby because they're pretty fragile.
No matter what type of gear you use to help monitor for invasive species, it is absolutely imperative that you thoroughly clean, drain, and dry all of your gear before entering another body of water. If we don't clean our gear, we could accidentally start a new infestation. Your effort to educate yourself on how to monitor your local watershed is key to protecting Montana's waters. Volunteers, just like you, have discovered and helped to contain the spread of aquatic invasive species in Montana. So your efforts, no matter how small, really do matter. In fact, we can't do it without you. For more information, you can visit fwp.mt.gov/AIS or contact the lab directly by calling 406-444-5299. Working together, we can all play a part in protecting Montana's waters.